Thank you. Good afternoon, everybody. I'm Gerald Zamponi. I'm the Senior Associate Dean for Research in the Cummings School of Medicine. I'm representing John Meddings, our Dean, who is unfortunately in Ottawa today, and I'm sure he wished he could be here. It's my pleasure to welcome you all to the 2016 Gairdner Foundation lectures. Okay, so every year the Gairdner Foundation's national program allows us to partake in a lecture series that takes place across the whole country where present and past laureates share their exciting work with the graduate students, faculty members, trainees, other researchers that we have in the various medical schools across the country. And it's we're very privileged that the Cummings School of Medicine is able to host that event again in 2016. So in our medical school, we place a lot of value and emphasis on science and research. And we're dedicated to being a national leader in these areas. And anyone who works in medicine and health sciences understands the value of research and the impact that it has on patients' health and our society in general. So today we will hear from two very incredible individuals about their work in vascular biology and on the CRISPR-Cas system, and the commitment to improving health and advancing science that Drs. Horvath and Dvorak display is going to inspire us all. So without further ado, and to open the lecture series, I'm going to welcome Dr. Ann Katzenberg, Associate Vice President for Research, to open the lectures. Thank you.
Thank you, Gerald, and good afternoon, everyone. It's a real pleasure to welcome you here today to honor the opening of the 2016 Gairdner Foundation Lectures. As you know, we're celebrating our 50th anniversary here at the University of Calgary, and 5 years ago, we set out with an ambitious plan to become one of Canada's leading research institutions to excel in research and innovative teaching methods. And today I can tell you that we're making tremendous progress toward our goal. And the Cummings School of Medicine is a major player in our success and our progress towards achieving excellence in research and innovative teaching methods. Our Cummings scholars conduct top quality research, and it's because of you that we're afforded the opportunity to showcase the world's best researchers through the Gairdner Foundation lectures. We're truly privileged to host these remarkable award-winning scholars here today. As you may know, many of the former Gairdner Award winners have gone on to win the Nobel Prize in either medicine or physiology. So to have them here to share their passion for research with us today is sure to be a memorable experience. This year, we're pleased to welcome Dr. Harold Dvorak, a Malandrat Distinguished Professor at the Beth Israel Deaconess Medical Center, Harvard University, and Dr. Philippe Horvath, a Senior Scientist with DuPont. You'll hear more about these fine scholars in a moment, and now I'll turn the podium over to Dr. Paul Coops.
So, thank you, everybody. Thank you. We're thrilled to have the recipient of the Canada Gairdner International Award for 2014, Dr. Harold Dvorak, here with us today. And I want to actually tell you about how the process works, since I chair the MRB, the Medical Research Board. We get about 160 applications each year of top-notch individuals, top-notch, and we send them out for review to 20 committee members, people that actually are very well established in their field. They review the applications and then rank them, and about the top 30 go to a second committee, the Medical Advisory Committee, made up of Gairdner Award winners, Nobel Prize winners. And these folks then debate which of the discoveries have made the largest impact on health and what are the most important discoveries. So the folks that end up at the very top are truly the very best in the world. So today, we're celebrating the discovery made by Dr. Harold Dvorak, vascular permeability factor, or as you cancer biologists know it, VEGF. And I just want to impress upon you the commitment that Dr. Dvorak has made to this field. He started more than 50 years ago studying permeability, the role of macrophage, the role of mast cells, the role of basophils, and the vasculature. He realized that there was a factor that made tumor vasculature quite permeable. He knew there was some other thing besides just histamine, for example. And in 1983, he published the discovery of vascular permeability factor. He went on to make some very, very important discoveries in this regard. And we now use anti-VEGF for cancers, for wet macular degeneration. We use it for diabetic retinopathy. So some very, very important diseases are now being treated based on that basic science discovery. Dr. Dvorak did his training at Princeton, his MD at Harvard, and then his internship at Mass General. He's the founding director of the Center for Vascular Biology Research at the Beth Israel Deaconess Medical Center, and he is the Mellencrot Distinguished Professor of Pathology at Harvard Medical School. He's won the Rue Whipple Award from the American Society for Investigative Pathology, the Le Follon de Lalande Grand Prix from the French Academy of Sciences, and the Gerotor Prize. These are all very, very highly regarded awards. And I know I speak for everyone at the Cummings School of Medicine when I say we're truly honored to have Dr. Dvorak here today. Everyone, Dr. Harold Dvorak.
See, I wonder if we can bring the slide up. Good, thank you. Well, it's a great honor to be here today. Even though I won the prize 2 years ago, it's very nice to be back. I've had a tour to Canada as a result of this. Once you're a Gairdner winner, as I was telling Philippe, you're not done with the foundation. You get to tour Canada. So I've been to St. John's, I've been to Saskatoon, I've been to Manitoba, I've been to Edmonton, and several other places. But I've not yet made it to the West Coast, so I'm hoping that they invite me next year. Anyways, so what I'd like to talk about is my title is up there. Just a very brief disclosure. So this is what I'd like to talk about. I'd like to talk about my Eureka Moment and the discovery of VEGF. I'd like to talk a little bit about angiogenesis and anti-angiogenesis, its current status, and then if I have time, going forward. So a long time ago, my wife and I a very long time ago, my wife and I were cellular immunologists, and we were looking at the immune response and particularly its histology. And you know the cellular immune response, and tuberculin reaction, response to viruses, contact allergens and allografts, and so on. And we made two unexpected discoveries that had not been made until that point. One is that basophils and mast cells were participating in these reactions, these T-cell mediated reactions. Of course, in those days, there was no such thing as a T-cell or a B-cell, but just lymphocytes, a long time ago. And so these are basophils here, lots of them. And then the other discovery we made was that fibrin was deposited in these reactions. We thought this was all part of the cell-mediated immune response. So my Eureka moment came a few years later. And we thought that tumors would make a good example of an antigen. And so we studied tumors as an antigen. But the surprising thing was, here you can see the tumor cells outlined. There's all this space in between them. And this was at 48 hours after a transplantable tumor, so long before there could be any possibility of immune response. This happened to be a guinea pig tumor, but it turns out to be responsive for a variety of others. What's in all this empty space here between the tumor cells? So if you take and this is was my Eureka moment. So if you look at higher power, what you see is tumor cells here, and you see what turned out to be fibrin here, and all this water and space in between. Remember that fibrin is a water-trapping gel. It's a gel. You know about Jell-O, and you know about other forms of gels that form. So it's a water-trapping matrix in which a small amount of protein holds together a lot of water. And so this is what you can see it here, and you can see it here if you clot fibrin in a test tube. And then, of course, if you do the usual immune reactions, you see the brown stuff. Here's the tumor is labeled with a T, and the brown stuff is fibrin. and this happens to be a guinea pig tumor, but down below is a human colon carcinoma the same way. And you can see that there's fibrin here, but there's also fibrous connective tissue here. And one of the questions is, what is the relationship between these two? So how do you get fibrin deposited in tissues? There's a long history of abnormalities of the clotting system in malignancy, particularly various types of leukemia, and many carcinomas as well. But for fibrin to be deposited outside the vasculature, two things have to take place. The vessels first have to be leaky so that plasma proteins can get out. And of course, the relevant plasma protein is fibrinogen, the plasma protein fibrinogen, and clotting proteins associated with it. And then together, these have to interact to induce extravascular clotting. So was this taking place in tumors? Well, the answer was yes. In the left panel, if you inject a fluorescinated dextran macromolecule dye intravenously and take a look at tumors, you see there's leakage. And so the tumor vessels are leaky. And they're not leaky universally, but they're leaky in hot spots, which is interesting and is something that still needs to be worked out in its entirety. In addition, there was clotting taking place, and the extrinsic clotting system was activated because of the fact that tumor cells on their surface express tissue factor. And further, tumor cells shed what we called microparticles, but now are called exosomes. And these little particles that are shed by the tumor cell, each of which expresses tissue factor on its surface. So that indeed, these requirements were fulfilled. You got extravascular, you got permeability and extravascular clotting taking place. So, hypothesis, obviously, what was going on? Tumors were secreting a unique vascular permeability factor, VPF, that was clearly degranulating mast cells and basophils. And here it is below my wife's beautiful pictures. Here's an intact basophil, and here's one that is degranulated. And so this is clearly what was going on. But then you do the experiment. And what we did was to take tumor cells and culture them in serum-free medium, and to take that serum-free medium and assess it for its ability to induce vascular permeability. And sure, there was a lot of it present. A lot of permeability-inducing activity was present. It was acting selectively on venules, just as histamine does. It was non-dialyzable and inactivated by heat and by proteases, and its synthesis was impaired by protein synthesis inhibitors, so it was clearly a protein. You see the very primitive methods we had. This is back in the late 1970s. But it didn't degranulate or release histamine from mast cells or basophils, and was not inhibited by antihistamines. So our hypothesis was wrong. Okay. VPF was acting directly on endothelial cells. So back to square one. And this is the work for which I received the Gairdner Prize. We purified it to homogeneity. This was long before the cloning days. We found that vascular permeability activity was very potent. It was about 50,000 times the potency of histamine on a molar basis. It was acting directly on vascular endothelium. An anti-VPF antibody blocked permeability and prevented ascites fluid accumulation, because ascites fluid, ascites tumors, result from the production of VPF and leakage of plasma proteins into the surrounding body cavities. And it was expressed by most malignant tumors. Here it is here. And you may be interested in the assay that was used. Here's this guinea pig, and he is the assay, pretty primitive assay. What you do is you inject what you think is a permeability column fraction into the guinea pig's skin like this, and you've injected a blue dye intravenously, such as Evans blue dye, a small molecule that attaches itself to plasma proteins. And so wherever the wherever albumin goes, the blue dye goes, and so you can assess column fractions in this way. It actually worked. Pretty primitive, however. Okay, so why is fibrin important? What does that have to do with anything? Well, it's important for a number of different reasons. First of all, it's a provisional stroma. Tumor cells, like other mammalian cells, can't swim in body fluids. They have to attach to things. And fibrin is a particularly good substrate for attachment. And it's very good because it's a promiscuous matrix for cell attachment and migration, because it binds to many different types of integrins And integrin is expressed, therefore, by many different cells, such as tumor cells, fibroblasts, endothelial cells, and so on. So it's very good for migrating. And you can illustrate this up here in vitro, where you have fibroblasts in a fibrin matrix. And they migrate through this fibrin matrix just as children do on a jungle gym. And they don't have to digest the matrix at all. They just migrate through it. But in addition, what was very interesting was the fact that fibrin itself is able to induce angiogenesis and fibroblast ingrowth, fibrous stromal connective tissue. And that's illustrated in this experiment here, a very simple experiment. Simply put a fibrin clot into the subcutaneous space of a guinea pig or a mouse, it doesn't matter. And this is the response that followed, a very rich angiogenic response with fibroblasts coming in. And eventually, this whole fibrin gel is replaced by fibrous connective tissue, just the kind of fibrous connective tissue that one sees in tumors, or as we'll see in wound healing. And the mechanisms by which this occurs is not entirely clear. It seems to induce the formation of growth factors locally. It sequesters growth factors, protecting them from proteolytic breakdown. And it has biologically active breakdown products. But the mechanisms by which this all takes place are not clear. But here you have this matrix, which is a provisional stroma, and then you have fibrin able to induce angiogenesis and able to induce fibrous connective tissue replacement. So this is the mechanism then of VEGF induces tumor stroma generation. You have increased vascular permeability, fibrinogen, other plasma proteins leak, you have extravascular clotting, the fibrin deposition as provisional stroma. Very interesting thing that takes place as well, that I don't have time to talk about today, is the plasma, when you have clotting taking place, plasma changes to serum with all sorts of growth factor release as well. So these events are very complex. Then you get angiogenesis, fibroblast infiltration, and over time, the fibrin provisional stroma is replaced by mature vascularized connective tissue. The result is that one looks at a human colon cancer, for example, or breast cancer, one sees primarily fibrous connective tissue with only traces of the fibrin the fibrous connective tissue replaced. Well, tumors don't invent anything. They simply make use of the toolkit provided them by the body, which the body has developed for other purposes. And so the events that tumors use to induce stroma are simply the wound healing response and the response to chronic inflammation. And this is illustrated in the left-hand panel here. Here is wound healing. And within just a few hours of a wound, this happens to be a skin wound, one sees a great upregulation, this is in situ hybridization, a great increase in VEGF production by the epidermis nearby. And this is triggered by the hypoxia induced by the wound itself. One gets leakage of the vessels are leaky. You get fibrin deposition, as you know. And the difference between a tumor and a wound is that in the case of the tumor, where VEGF production is turned on by a variety of factors, genetic changes, in addition to hypoxia, in the case of the wound, the VEGF production goes down over time, and the vascular permeability goes down over time in parallel as the wound is healed and as vascularization is reestablished and the hypoxia goes away. The same series of events takes place in a variety of chronic inflammatory diseases, such as rheumatoid arthritis, psoriasis, and so on. And here again, one has fibrin deposition, VEGF production. The pannus has a lot of fibrin deposition in it. And again, there's fibrous connective tissue replacement of that fibrin over time. So we coined the expression that tumors are wounds that do not heal. Obviously, this is not entirely true. There's a lot more to tumors than being a wound that doesn't heal. But these same bodily processes that take place in tumors are taking place in tumors and wounds and chronic inflammatory diseases. Well, I'm going to shift gears a little bit at this point, and my friend Judah Folkman, back in 1971, gave a talk at our hospital, Beth Israel. It was then the Beth Israel Hospital. And he postulated three things. His work was published in the New England Journal. And he said that tumors have to induce new blood vessels to grow beyond minimal size. He said that tumors were making a tumor angiogenic factor. He called it a TAF. Well, that turned out to be VEGF. and that anti-angiogenesis should be effective therapy for a variety of reasons. One is if you kill one blood vessel, you should kill a zillion tumor cells, all the tumor cells supplied by that vessel. Tumor vessels are abnormal, so you should be able to attack them. It should be applicable to many different cancers, because presumably many different types of cancer were making the same angiogenic factor. And the drug should be very accessible because you're attacking tumor lining blood vessels, endothelial cells, so there should be no trouble with access. So what about tumor angiogenesis now in 2016? Well, Folkman's postulates have been largely validated. Tumors generally do have to induce new blood vessels if they're going to grow beyond minimal size, although that's not always the case. And they do so at least initially by secreting VPF, that was Folkman's postulated TAF. Indeed, tumor vessels are abnormal, and so they are potentially targetable. And anti-VEGF therapy has certainly found a place in treating human cancer. But unfortunately, the promise of anti-VEGF therapy has really not been realized in cancer. And the question is, why is that? Certainly one can say that anti-VEGF therapy, and whether he's using an antibody to VEGF or a VEGF trap or a receptor tyrosine kinase inhibitor, for whatever reason, you do indeed prevent the growth of many fast-growing mouse tumors. But of course, mouse tumors are not human tumors. But it's less effective on slowly growing mouse tumors or tumors that have established their vasculature. And as we all know, it has limited effectiveness in cancer patients. The original effect was in colon cancer, shown here, where there was when it was given along with 3 types of chemotherapy, it extended life by only 4 to 5 months on a mean average. Now, some patients benefited a lot more, but for the most part, the average, and some didn't respond at all. And it's ineffective for a variety of other cancers. And so some people have actually said, well, it's really of limited benefit, high cost, maybe it should be abandoned. But they are highly profitable. Avastin sales, the antibody against VEGF made by Genentech, generates sales of more than $6 billion a year. So maybe it's a good time to ask some basic questions. What are tumor vessels? How do they form? Why doesn't anti-VEGF therapy work better? And if I have time, can anti-angiogenesis therapy improve? Okay, so it has often been thought that tumor vessels were a single type, that angiogenesis led to the formation of angiogenic vessels of a single type. That's not true. Tumor vessels are extremely heterogeneous and have at least 6 different types. And I've shown them down here, I won't go into it, but most indeed are abnormal and do differ significantly from those found in normal tissues. And a point I want to emphasize is not only is there angiogenesis, that is the growth of small blood vessels, but also arteriovenogenesis, growth of new arteries and new veins that supply and drain the tumor. So tumor vessels are heterogeneous, not of a single type. How do tumor vessels form? Well, they form by making VEGF. The tumor vasculature is hard to study in situ because they're a minority component. There's 6 different types, and you can't tell the steps and mechanisms by which each forms. You can't tell the formation sequence. You can't isolate each different tumor vessel type as you'd like to, and sufficient numbers for molecular characterization. So what we did is took a reductionist approach, and we expressed new blood vessels that were induced with an adenovirus expressing VEGF. So in fact, what we found was that AdVEGF, an adenovirus expressing VEGF, the mouse form, induces all 6 different types of tumor blood vessels, and it does so in sequence and in large numbers in nude mice. And a nice thing about this assay is that one can do it in the mouse ear and follow it. So here's a mouse ear at day 1 after the injection of an adenovirus expressing VEGF, and here it progresses over time. You see you get lots and lots of angiogenesis. And here, 128 days later, you still have activity going on. You still have large numbers of abnormal vessels that have formed. So this is the system we've used. And so if you have VEGF or made by an adenovirus or by a tumor, you get angiogenesis with the formation of something called a mother vessel, and these go on to other types of vessels. You also get arteriogenesis, and you also get venogenesis. But I'd like to spend a moment talking about mother vessels, the first angiogenic blood vessel to form. And you'll see the reasons for this. This is the mother vessel right here, formed from a preceding venial or capillary. And this is what these mother vessels look like. Here at the light microscopic level, you start with a capillary, and you get this very large vessel that is formed, 4 to 5-fold increase in cross-sectional area. And here, by electron microscopy, you see the vessel, the venial, and here's this very much larger vessel that is formed. So they're very large, they're hyperpermeable, they're very thinned endothelium, pericytes have detached, pericytes up here have detached, and there's endothelial cell division going on. These are the properties that a mother vessel has. So how do you get a mother vessel? Well, the first thing that has to happen is the basal membrane has to be digested. The basal membranes surrounding small blood vessels are very rigid structures. And if you were to try to attempt to enlarge those vessels simply by increasing internal vascular pressure, you could get about a 30% increase and no more. And beyond that, the vessel would actually burst. So to get this very large 4 to 5-fold, 400 to 500-fold increase, you really have to have digestion of these basal membranes. And we've worked out the systems by which this takes place. When that happens, the pericytes, the pericytes fall off, leaving the endothelial cells as the sole lining cell. So here you can see the pericyte has fallen off, this very large vessel with broken down basal membrane. And when these events have taken place, simple hydrostatic pressure drives 4 to 5-fold vessel enlargement with endothelial cell thinning. Now, why is mother vessels, why are they of interest? Well, they're not only the first vessels that form in tumors, they're also the first vessels that form in healing wounds and also in chronic inflammatory diseases, work done by others. And of course, when we're talking about wounds, we're not talking about a simple cut in the finger. We're talking about wounds such as myocardial infarcts, shown here, or strokes, or other examples of tissue damage. And of all the 6 types of vessels, these are the only type that is permeable. These are the types of vessels that are permeable. Normal vessels of other types induced by VEGF are not permeable, but mother vessels are permeable. So, given these vessels and so on, why doesn't anti-VEGF therapy work better? And people have talked about this for a long time. Maybe we need to treat better. That was Judah Folkman's idea, was that better dosing would be important. And we're dealing with sick patients, not dealing with healthy mice that are used in the laboratory, and there's a difference. One big point that has been made is that ischemia induced by killing some of the tumor vessels, but not all, leads to ischemic tumor cells, and ischemic tumor cells make more VEGF and make more of other growth factors. And some have taken the response, well, Folkman's idea was simply wrong. Antiangiogenesis is of limited value, and you're not going to be able to starve tumors. But there's 3 other possibilities that deserve consideration. The first of these is that tumors co-opt pre-existing vessels. In other words, they don't have to induce the formation of new vessels. They use the ones that are present. Another is that some tumors are able to survive hypoxia. They're able to, they live on the edge. They don't need much in the way of blood vessels. And finally, not all tumor blood vessels are equally susceptible to anti-VEGF therapy. Remember, I mentioned there's 6 different types. Maybe they don't all need, they won't all respond. So let's take a look at these 3 possibilities. Possibility number 1 is that cooption, use of normal vessels that are present. And this certainly is the case of tumor cells that have metastasized to the lung. Here's the commonly used B16 melanoma sitting in the lung, a huge amount of tumor, and yet all the vessels around it are normal. There are no new blood vessels that form. And this makes a lot of sense if you think about it, because why would a tumor want to induce new blood vessels if it's sitting there in the lung? I mean, it's tumor cell heaven. It's living in a place where there's an enormous amount of oxygen, and the capillaries of the lung supply all the nutrients and take away all the waste products. Many human tumors are in the same situation. This happens to be a human pancreatic carcinoma, metastatic to the lung. But some other metastatic human tumors do, in fact, induce angiogenesis. So that's one possibility, cooption. Another possibility is that there are some tumors that don't require much in the way of new blood vessels. They are able to live under starved conditions, and This is certainly true of some breast cancers and some pancreatic cancers. They just sit there, and in their semi-starved way, they continue to live. They don't do much, but they don't need new blood vessels, or very little in the way of new blood vessels. And then there's a third possibility, which is the one we've been interested in, and that is that anti-VEGF or anti-VEGF receptor therapy affects only some of the 6 different types of tumor vessels. As I tried to indicate, tumor vessels form linearly. You have early vessels, and they evolve into late vessels over time. And in mice, as I mentioned, anti-VEGF therapy is very good at dealing with early tumor vessels, but apparently less effective in established tumors that have many late vessels. And this, of course, is the case of human tumors, where human tumors have been around a long time before diagnosis is made, and months or years. And so one might expect that blood vessels have been forming for a long period of time, and that the majority of them will be of the late type. So our hypothesis is early vessels are VEGF dependent, whereas late vessels, although they were induced by VEGF, have lost dependency on tumor VEGF, and they're no longer susceptible to anti-VEGF therapy. So to test that hypothesis, we did this experiment, and it's a complicated slide, but I call your attention only to this area up here. Here is the control, where we gave a control substance, and you can see the tumor vessels evolve over time and continuously form. Here is treatment with an anti-VEGF drug, and one can see that the early vessels are blocked up to about 21 days of their formation. But after that point, they're unaffected. They don't differ really from control vessels. So in fact, it is true that the early vessels are very susceptible to the anti-VEGF therapy. The late vessels are relatively or even completely insensitive to therapy. So I'm nearly done. I think I have time. The last question I wanted to address with you is, can anti-angiogenesis treatment improve? Well, you can't do much about cooption, or tumors that can survive with limited vasculature. You can attempt to target additional growth factors and receptors besides VEGF receptor and its receptor. This has been tried, but it hasn't been very successful. So the alternative is to find new targets, and especially on the feeding arteries. If this is the complex where you have feeding arteries and you have angiogenic blood vessels and draining veins, if you were a plumber, where would you go? Would you, if this were a house that you wanted to turn off the water on, would you go to every faucet in the kitchen and the bedrooms and so on? No, you'd go to where the water comes into the house, and you'd aim, in other words, for the feeding arteries. And so that is what we've attempted to do. And our strategy has been to screen for new targets in these late VEGF-induced blood vessels of this type. And the target we've found and are working on a little bit is something called transmembrane 4L6 family 1, TM4SF1. And this is a antigen, actually, that the Hellstrom group discovered in the 1980s and 90s. And they found it as a tumor cell target, and to their disappointment, they found it was also expressed at low levels by normal vascular endothelial cells. And they went through various hoops with antibodies, and they developed some activity against late-stage cancer patients. But these were the days when you did not humanize the antibody, and that they attached I-131 to the antibody, and that had all sorts of toxic effects. So a young lady who's working with me, Shuxing Shi Jamanet, took up this project again, and she found that although it did indeed have weak expression on normal tumors, on normal vascular endothelial cells, it was strongly expressed on cultured vascular endothelial cells, and strongly expressed on the endothelial cells supplying tumors. In vitro, one could knock it down and block endothelial cell proliferation, motility, and in fact, the knockdown induces senescence. And in vivo, the knockout is embryonic lethal due to a failure of blood vessel development. So she went on and has found that it's actually very effective. An antibody-bearing an antibody-drug conjugate is very effective in treating a wide variety of tumors, getting complete regressions, not in all tumors, but in a wide variety of tumors. And the effectiveness seems to be roughly correlated with the expression levels of TM4 on the tumor cells and on the blood vessels. So here you have a target that is dually expressed on tumor cells and on blood vessels, and it seems to have some promise. So take-home message. All tumors have stroma. Tumors are not just a bag of tumor cells. Tumors generate new stroma in blood vessels by secreting VEGF. Vascular permeability has a key role in initiating angiogenesis and stroma formation, and fibrin has a very important role in that process. It induces initial provisional stroma and stimulates the formation of mature stroma. And tumor stroma formation mimics wound healing and chronic inflammation. Tumor vessels are heterogeneous of at least 6 different types and 2 distinct processes: angiogenesis, formation of small vessels, and arteriovenogenesis, the formation of large ones. Mother vessels are the first new angiogenic blood vessel to form, and this is the only part of the tumor vasculature that is hyperpermeable to proteins. Anti-VEGF therapy prevents angiogenesis and arteriovenogenesis, but it only regresses early vessels. It doesn't regress late angiogenic vessels induced later. And finally, new vascular targets are needed. TM4SF1 is one attractive candidate, but there may be others that need to be looked at. And so I thank you for your attention. Any questions? Over here. Over there.
There's a key partner with your VEGF that relates to the fibrin deposition, which is also a vascular permeability factor called thrombin. And so my question is, can you talk about the relative contributions, early, late vessels, and the contribution of, well, the coagulation pathway itself, including thrombin?
Yeah. So thrombin is a permeability factor in some species, but not in others. It has many other properties acting on endothelial cells, but not inducing permeability, for example, in the guinea pig or humans.
Thanks very much. I have a question about endothelial cell proliferation. I'm not sure if you actually specifically addressed that, but I think you were talking around that.
Yep.
Like the turnover in quiescent endothelium is very low, like less than a percentage point. And you get into tumors,
Right.
It does change and it's higher. So is that what you're also referring to? Like there's variability in the proliferation index that may affect the targeted therapies that you might develop?
Yeah. So, I mean, one of the questions that I hope you all heard the question is the endothelial cells in tumors are dividing. The endothelial cells in wounds are dividing and so on, chronic inflammation, but not otherwise. So are these drugs having an effect on the proliferative effect? Some people have thought that actually chemotherapy is acting, at least to some extent, in this way. Whether the anti-VEGF therapy is acting on proliferation or not, I don't know. But it is indeed the proliferative vessels that are effective, the mother vessels. They are the proliferative vessels. So it is these vessels that are sensitive to anti-VEGF therapy.